but in a more relaxed term, everyone knows gene expression is on the title regulation, but the actual picture is there's not much knowledge when we talk about any particular mutation with disease, because most of focus are in the coding region. Everyone talk about a mutation. It's coding some protein truncated, starved, blah, blah, blah. How about the regulator area? It's really a serious challenge. As I said yesterday, current technology by no means provide a de novo determination for any genetic disorder in the regulated area. We really need to have some very powerful genome-wide tool to address this issue. And from our preliminary data work on breast cancer, I strongly believe the regulated area contain a rich information to address basic biological issue and also medical issue, and particularly in the cancer. No matter how much effort people make nowadays on cancer genetics, cancer genomics, not much about regulated area. That's highly unlikely. And in fact, when we talk about the cancer in general, we talk about the gene misfunction cause cancer. You look at all genome-wide study. Most of them are not very as expected. Say the large-scale cancer genome study. It really require use the sample size of tens of thousands in order to identify some mutation in the coding area with statistic significance. And we know every cancer, now they talk about personal medicine, talk about the precision medicine. You don't expect that so many things happen in random, say a genome, in somatic genome in cancer. I don't think there is any high chance to identify as a very common cause for a particular type of cancer. That's already proved by all genome study. Whenever identify some common mutation in the coding area, normally you see very low percentage of cases contain the same mutation. You not talk about the same position in the coding area. The thing about the coding gene only account for 1% of the genome. 99% of the genome are not coding for anything, but a part of them are regulated area, say about 2-3%. Even this 2-3%, there's no systematic analysis so far. Why? The tool. Really, there's no tool. That's why I'm suggesting, should we focus on more in the regulated area for particular disease study? And I ask for your comments. Anyone make comments for this? What do you think about it? That's the whole point about the workshop, right? And that professor, yesterday you talked about the polymer, use polymer mapping promoter regulated area. Do you have any comments for that? Can push your system become a universal general tool for anyone interested in analyze regulated area?
Depends on how much data you want to look at. I mean, the problem with doing database searches is you get a large number of positives. And so you end up in database searching, constantly setting windows on, it has to be this significance or I won't look at it. I have to have sequences much or I won't look at it. But there's a huge amount of data there.
Your system work on 384 ones, right?
Yeah, well, that's how many wells are on the plate.
Will it be possible to push go to like array system with genome-wide coverage?
Yes, it would. I mean, essentially, well, the other limit to it is how much essentially the density phenomenon. I was discussing this with a colleague yesterday, that you have to have it's about 10,000 ions before you can detect it in a mass spectrometer. And so you have to have a spot of a size that would contain 10,000 ions, okay? And so you could make a large array, maybe spot 20 nanoliters instead of spotting 2 microliters like we did. And, as long as there's 10,000 ions there, you would detect it, and you would be able to do analysis on it.
So this is a radical possibility. Practically, can you push go that direction?
Well, graduate students are only in the lab for 4 years.
Can Affymetrix get involved, adapt to their system, high spot system?
Well, there are other methods than mine. I mean, for example, CELD, which is where you covalently attach a DNA to a surface. Okay, that essentially in that system, the problem is you can have a very dense probe, which for DNA binds anything if you have a high enough amount, right? I mean, transcription factors bind to DNA with nanomolar affinity, but when you put a huge amount of DNA on a two-dimensional surface, concentration's undefined, right? It's just concentration's three-dimensional. How do you define it for 2 dimensions?
You guys think about whole genome sequence is a way to go to analyze a regular area? Now it's much cheaper, and the lowest one whole genome sequence is about $1,300 whole genome.
Yeah, the biggest problem there is the quality of software for identifying binding sites, which was a point I made in a question earlier. I mean, we ended up doing one project that spent about 2 years where we were looking at a promoter it's involved in HIV, and the computer insisted there was an NF-kappa B site there. There is no NF-kappa B that binds to that DNA. So a lot of what you get out of a computer, it's kind of like doing a prosite search and believing that everything is myristylated or everything's phosphorylated. Computers predict things that aren't real.
Yeah, but now the DNA sequencing technology, particularly for next-gen sequencing, obviously the first lines create a huge amount of digital information. How to mine the data, get the value from the massive data set. Obviously, computer is essential tool. Without the computer, without bioinformatics, when rarely cannot do anything.
Yeah, it's kind of like the old saying they used to have with computers: garbage in, garbage out. Right? I mean, you essentially get what you're looking for in computers.
Yeah.
And you have to be aware of that. That's the only caveat, okay? Computers are wonderful tools. I certainly couldn't do proteomics without them. But the thing is, if it tells me that I have actin in a sample and the sample should not have actin in it, I tend to go with my own beliefs. I'll probably go ahead and look for actin, get an antibody and do a Western blot, but I'm pretty sure the computer is telling me something that's not accurate.
Yeah. And other people comments about that?
The thing about promoters, though, is you already know enough to be able to identify a large number of them, right? I mean, there are 23,000 genes, so there must be minimally 23,000 promoters.
Far more than this.
Oh, there would be more, because there are a lot of transcripts that have alternate promoter usage.
Yeah.
And promoters can occur in introns as well as in exons. So you know there's 23,000 minimum. But you also know a lot of characteristics of promoters. They have ta-ta boxes, they have INRs, they have DPE sequences, BRE sequences, right? So you do know what a promoter looks like. And I think by combining all of those things that you know, right? I mean, if you look at an intron and you find a ta-ta box and a BRE sequence, I'd consider that a possible promoter.
Yeah, but for promoter, for ta-ta box, if the gene has ta-ta box, we'll suggest this is promoter. But any ta-ta box sequence, if it looks like ta-ta box, is located nowhere in the middle of the genic region. There's no indication those are ta-ta box. That's a trivial part for computational prediction. They always pick up a lot of junk. They say this is ta-ta box. But in fact, that's nothing to do. It's just by chance with TAA.
Yeah, that's why I'm saying it has to be combinatorial, right? I mean, ta-ta boxes, you know, it's very rare you ever see a TAA for a ta-ta box. They don't look like that. Quite often, that second T is a C, for example, in ta-ta boxes, okay? And INR sequences are not completely defined either. I mean, they begin with an HE, essentially. Other than that, they're quite variable, okay? Same thing with BREs. I mean, BRE sequences are not clearly identified all the time. But if a computer can find many of those things, I would say that is a good candidate for further investigation.
Mm-hmm. Can you say something about how variation in promote structure affect a real specific expression? A real specific expression. Now you see we have non-range sequencing. We can get half type. This is no problem. So now my question is, you see, you study the variation in promoted area, promoted structure. How variation in promoted structure will affect a real specific expression. So I observe, you see, a real specific expression. The also, you see, there we can observe variation across the gene. Different genomic position, we observe different real specific expression. I don't understand. It should be the same, right?
Well, if my understanding, if in the regular area, particularly for some particular transcription binding sites motif, if there's any significant change for some highly conserved base, theoretically this should affect the expression level, either increase or decrease. And then this is, in fact, is theoretically same as the basic fundamental biology quantitative expression, right? Dosage effects. And the thing about the network issue, every gene expressed not only as yes or no, but how much. If the quantitative feature change, one of the key gene, if there's downstream 10 targets, that's obviously affect all network combination.
I mean, you see, idea specific. You see, the same genomic position, two different area, you will have different expression.
You mean different organ, different tissue?
Idea
okay.
Specific expression.
Yeah.
Okay. And then there are not two questions. Question one, you see, you observe idea, different idea, we have different expression on the same genomic position. Second, different genomic position. Now, we have different idea specific expression. Now, someone is just assume and the same haplotype. These should have the same expression level. However, you see, I not with a lot of data, I found, you see, there are no common expression level for the allele specific expression. Now, maybe someone shall get this caused by sequencing technology variation.
But that's a technical
However,
issue.
now we have long-range sequencing. We can sequence haplotype. Now just shotgun sequencing. So maybe now someone do some experiments, examine. So I might know gene negation. Many say I don't believe. Then my question is, you should observe data. You should explain the data. You not just the hypothesis. You're a biologist. Your hypothesis we should examine by experiments.
Well, first of all, there's a technical limitation. I think if there's 20,000 genes, you see 100 gene promoter variation. The benchmark can only adjust one by one. That's a much simplified system. Theoretically, I was thinking this is a network. This 100 gene affect the 20,000 network. And experimentally, I don't think it can prove this at this stage on the technical limitation. On the other side, theoretical biologists or computational biologists, they can model in this system. You saw a network, beautiful network picture, but neither, I don't believe it. How to prove this, right? That's the caveat of the current science. Biology is limited, very traditional way, one gene, one function, one story. In the real field, in the real world, it doesn't exist. But on the other side, the theoretical biologists always model something which is not based on experimental data because there's no data. And then use existing theory, Bayesian theory, for example. But this can explain everything. Like particularly for transcription factor mutation network, you can see easily nowadays Nature paper, science paper. Look, the picture are so nice, like artwork. I don't believe it because there's no way to prove this. But this is current art of the science. Anyone have comments about that?
But a lot of times what those pictures are, aren't they, is a dramatic way of showing a hypothesis. And perhaps that is the complaint you're making, that it's hypothetical, that there's no data yet to support that, right?
Yeah.
I mean, we all have to do things like table of contents, illustrations, that we're trying to make one picture to say what we were doing, right? I just did one for a paper on the method I talked about yesterday. And I essentially showed a promoter and an open reading frame, transcription factors binding, and underneath it I put question marks for where they bind, right? I mean, you know, does that represent the experiment? Yes, it represents the experiment. But if somebody takes that too literally, then they're going to be misled. They're going to think transcription factors are little color balloons and question marks exist in DNA, right? So we're trying to make a hypothesis that we can test. That's all the pictures on covers are.
But my thought is, in many situations, there's no way use current benchwork style to prove this kind of highly theoretical model. Very hard to prove this.
Yeah, I guess a lot of times we just stumble on things. I mean, like, I've worked on CEBP for a very long time, and Dr. Liu informed me yesterday that it can be a tumor suppressor, right? That 3' untranslated region. And I doubt seriously that he went into those studies expecting to find a tumor suppressor and a transcription factor, right? And so we're lucky sometimes.
Mm.
Right, I mean, you know, Fleming didn't go looking for penicillin. Penicillin came and found him.
Well, for that matter, Newton wasn't looking for apples, right?
Do what?
Newton wasn't looking for apples.
Yeah, he wasn't looking for apples. He was a microbiologist after all.
I'm not sure if you're asking a specific question, but one of the important points of interest, in my opinion, is that this is very common in other sciences, but in biology and to a less extent chemistry is lacking, I think. And it is the ability to, it's a complex systems issue, it's the ability to integrate data from across fields, across scales, and across time, so that you can put together things that do not seem related, but they are indeed related through complex chains of interaction. So I think that's a very important aspect, particularly in genetics and understanding the notion of variation that you were raising. So how can we come up with a framework in which is in some sense universal, in the sense that any relationships that might exist become evident and are revealed outside the specific context in which they may arise. So I think that's a very important problem to solve, and one that I think might yield some interesting dividends. So integrating across scales in space and time is extremely important, I think.
Yeah, I fully agree. My argument is the current benchwork technology are based on a reductionist. Simplify everything to find one key element, control everything. But in the real world, it's not the case. Right? Particularly dealing with genomics. Every time you have data set, I see hundreds of mutations. How can I simplify the system, find the one I believe is the major player, but in fact it may not be? I can make the story nice Western, right? Change. It doesn't mean it is the cause or the consequence.
Yeah, but that's just the point of complex systems. Complex systems research takes the opposite view. It takes the sense that I learn more from a complex model of a very simple phenomenon that I do from a very simple explanation to a very complex phenomenon. So it's the opposite exactly. So this is a very different approach in complex systems than it is in reductionism.
Yeah, how to combine the multi-layered rich data with the reduction theory? That's obviously a very big challenge. I don't see there's any powerful solution right now. People go opposite direction, try to combine. It's not on the solid base. That's my feeling. Yeah.
One of the points in complex systems is that things are so complicated. I mean, the human mind can only process. We are essentially sequential processors when it comes to, abstract thinking. We can only think of 1, 2, 3 things at a time in our heads. And there are psychological rules that have been well researched and tell you the limitations. Whereas in the phenomena that we're interested in here, we have hundreds of things, thousands of things going on at once. So as you know, we may not trust the computer entirely, and we shouldn't, but computers will have the ability to handle all this parallelism and tell us something interesting that we may not have been able to see through there are limitations to the kind of analysis we can do, and I don't think we'll be able to continue without any more powerful device that will allow us to handle all that parallelism all at once. That we focus on specific regulators and enhancers and so forth. But is the dynamics of the things coming together that control or determine most of the outcomes, not just one or two or three at a time.
Yeah, that's what we can do right now. I hope there's some revolution happens to break this barrier between theoretical and the practical system and become universal approach. Yeah. No more? No more comments? All right, thanks.